The overall goal of this procedure is to precisely analyze the amount and the structure of each Sphingomyelin species in biological samples.
This method can help answer key questions in the lipid field pertaining to lipid biology and lipid metabolism. The main advantage of this technique is that we can characterize a variety of Sphingomyelin species in biological samples by liquid chromatography mass spectrometry system.
To begin, remove the culture media from a 10 cm tissue culture dish containing HeLa cells and rinse the cells twice with 6 ml of ice-cold PBS. Then, add 1 ml of the ice-cold PBS into the 10 cm tissue culture dish and use a cell scraper to harvest the cells. Once removed from the dish's surface, transfer the cells into 2 ml siliconized plastic tubes. Following centrifugation, remove the supernatant and add 1 ml of methanol to each cell pellet. Then briefly vortex the tubes. Next, place the samples into a bath-type sonicator and sonicate at 200 watts for 5 minutes. When finished, transfer the whole cell homogenate into test tubes equipped with Teflon-lined screw caps. Now, add 1 ml of methanol, 1 ml of chloroform, 0.8 ml of double distilled water, and 50 microliters of 10 micromolar of an internal standard into each test tube. Cap the tubes and vortex them vigorously at 2,500 RPM for 5 minutes at room temperature. Then add an additional 1 ml of chloroform and 1 ml of double distilled water to each tube. Again, vortex the tubes vigorously at 2,500 RPM for 5 minutes at room temperature.